Today our topic is ICQ-1 Stability Guidelines. The ICQ-1 Guidelines, also known as the ICQ-1A R2 Guideline, provide recommendations for conducting stability testing of new drug substances and products. These guidelines, developed by the International Council for Harmonization of Technical Requirements for Pharmaceuticals for Human Use, IC, outline the principles and practices for assessing the stability of pharmaceutical products. It covers aspects like testing conditions, sampling, storage conditions, and data interpretation to ensure the quality and reliability of stability data. Conducting stability testing involves subjecting drug substances or products to controlled conditions to evaluate their stability over time. Here's a basic overview of the process. Selection of batches. Choose representative batches of the drug substance or product for testing, considering different strengths, formulations, and packaging options. Storage conditions. Determine the appropriate storage conditions for testing. This can include different temperatures, e.g., room temperature, accelerated conditions, refrigeration and freezing, humidity levels, and exposure to light. Time points. Define specific time points for testing, which may range from a few weeks to several years, depending on the intended shelf life. Sampling. At each time point, take samples from the batches. The number of samples and their frequency depends on regulatory guidelines and the study design. Analytical testing. Perform various tests to evaluate the physical, chemical, and microbiological properties of the samples. This could include tests for degradation products, potency, impurities, and appearance. Data analysis. Analyze the test results to assess the stability of the drug substance or product. Compare the results to the acceptance criteria defined in regulatory guidelines. Extrapolation. Based on the data, extrapolate the stability of the drug substance or product beyond the actual testing duration. This helps determine its shelf life under real-world conditions. Documentation. Document all testing procedures, results, and conclusions in a stability report. This report is submitted to regulatory authorities as part of the drug approval process. Ongoing monitoring. After the initial stability testing, continue to monitor the product in real-time conditions throughout its shelf life to confirm the accuracy of the extrapolated data. It's important to follow established regulatory guidelines, such as ICQ-1, to ensure that the stability testing is conducted accurately and consistently. The ultimate goal is to provide reliable data on how the product's quality, safety, and efficacy change over time, enabling manufacturers to establish appropriate storage conditions and expiration dates.
Now we'll discuss about the ICS stability zones, and it is one of the most important slides of today's presentation. So the globe has been divided into 4 climate zones based on the mean kinetic temperature. So the zone 1 basically temperate zone, zone 2 is Mediterranean zone, zone 3 is a hot and dry zone, zone 4A hot humid zone, and zone 4B hot very humid zone. Now a very important point to note here that the zones has been divided based on the mean kinetic temperature. So a mean kinetic temperature, in a simple language, it is nothing but a temperature which is very close to mean temperature for that particular region. Now if I have to give you an example of this, take example of India. Now in India, in summer there are regions where the temperature goes above 45 degrees centigrade, and in winter there are places where the temperature falls below 10 degrees centigrade. So it is not constant. The temperature, it varies over time around the year. But now if you see the mean kinetic temperature for the zone 4B, it is 3075. That means that if you keep your samples in this 3075 degrees condition over a long period of time, it is going to have the same thermal challenge as would have been experienced over a range of both higher and lower temperature. So this is basically a mean kinetic temperature. Now let's look at the map illustrations of ICS zone. So there is a global map you can see on the screen. The upper regions of the map, which is blue in color, shows the ICS zone 1. Now the regions below that, which is in green color, shows the zone 2 regions, and below that the orange color one shows the zone 3, followed by the red color regions which shows the ICS zone 4. Now let's look at the regulatory requirements for drug substance and products intended to be stored at room temperature. So as I have mentioned in my last video, there are basically 3 kinds of stability studies: long-term, intermediate, and accelerated. So in general case where you are going to store your product in the room temperature, the long-term testing condition would be 25 degrees centigrade and 60% RH plus minus 5% RH, or you can do it at 30 degree followed by 65% RH. If you're going for intermediate, it would be 30-65, and if you're going for accelerated study, it would be 40 degrees centigrade plus minus 2 followed by 70% relative humidity plus minus 5% relative humidity. Now, one thing that you have to keep in mind is that whenever you are submitting your dossier, a minimum of 12-month long-term data has to be submitted along with a 6-month accelerated data. And in case if you are submitting intermediate data, a 6th month data, intermediate stability study data has to be submitted. This is the minimum requirement. Without this, your dossier won't be accepted for review. Now, if you look at the testing frequencies, this is one of the very important slide. Let's go one by one. So for the long-term stability studies, for the first year, the testing frequencies would be 3 months interval. That means 0 month, 3 months, 6 month, 9 month, followed by 12 month. For the second year, it will be 6-month interval. That means 18 followed by 24. And after that, it would be annual. That means 36. Accelerated stability study, it should be done for 6 months with a 3 months interval. That means 0, 3 and 6. And for the intermediate stability study, it has to be given for 1 year and a minimum of 4 time points, that is 0, 6